Okay, protein synthesis. This is the process wherein you make, or the body can make proteins under the direction of DNA molecules. So, for this process, the body is making proteins in the form of such as your skin, your hair, the enzymes in your bodies, some hormones and other proteins. Okay, so the summary or overview of this process is this structure. Okay, so protein synthesis starts with the replication of a DNA, because DNA is very important in protein synthesis. And once DNA has been replicated, this undergoes transcription process. Okay, so transcription process, which happens inside the nucleus, such that the DNA will be transcribed into an mRNA. After being transcribed, the mRNA will go out of the cell nucleus, going into cytoplasm for a translation to occur. And translation is a process wherein mRNA will be coding or will be directing the protein synthesis. So we start with replication, but the main part of protein synthesis is transcription and translation. So, laging tatandaan, transcription, see first before the translation. And there are many RNA involved in this process. So, we have during the transcription, those involved RNA are hnRNA or heterogeneous nuclear RNA, snRNA or small nuclear RNA, mRNA or the messenger RNA, and during translation, RNA involves are mRNA, rRNA, which is ribosomal RNA, and tRNA, which stands for transfer RNA. Okay, so always bear in mind replication. The DNA, the DNA creates an exact copy of itself, and then after that, transcription, the DNA will be transcribing to produce mRNA, and translation is mRNA is directing the protein or the creation of protein or protein synthesis. Now, before we proceed to the details of these processes, let's first have a recap of the comparison of DNA and RNA kasi na present sila dito sa mga processes na to. So, the sugar unit in the backbone of RNA is ribose. Okay, and the sugar in the DNA is deoxyribose. Okay, so we should be clear on that. RNA has ribose, DNA has sugar ribose in the sugar unit backbone, the sugar unit in the backbone of those nucleic acid. Now, for in terms of bases, the DNA will only have um, adenine paired with thymine and guanine being paired with cytosine. However, in RNA, this thymine is replaced by uracil. So, adenine base paired with uracil and guanine still being paired with cytosine. Okay, so still this base pairing is due to the hydrogen bond attraction between these bases. Okay. Then, RNA is a single-strand molecule, so isang strand lang siya, while as DNA is a double-stranded nucleic acid. Okay? RNA contains an equal amount of specific bases, while DNA, we know that the percentage of adenine will be equal to the percentage of thymine, and guanine will be equal to the percentage of cytosine because of the base pairing. Okay? Pero kay RNA, hindi equal. Kasi nga, isang strand lang siya. Okay? And then we have RNA to be a much smaller molecule than DNA kasi single strand lang siya and DNA is a double-stranded molecule kaya mas maliit siya. Mas maliit si RNA. Now, before again we proceed to the details or details of transcription, we have to review the characteristics of the 5 types of RNA molecules kasi involved sila dito sa protein synthesis. When we say hnRNA, that is heterogeneous nucleic acid, heterogeneous ibig sabihin hindi uniform. Okay? This RNA is directly produced via DNA transcription. So ito yung product ng DNA transcription. mRNA or messenger from the name itself, message. So it carries an instruction for protein synthesis, and that instruction is eventually the genetic information, okay, that is required for the protein synthesis. Now we have small nuclear RNA or snRNA, which facilitates the conversion of this hnRNA into an mRNA. So, siyang magko-convert ng heterogeneous RNA to become a messenger RNA. So, that's the function of snRNA. We also have ribosomal RNA or rRNA. This combines with some or with specific proteins para ma-produce ang ribosomes. We know that ribosome is part of the cell. Usually sila yung mga studs dun sa rough endoplasmic reticulum. And we have transfer RNA or tRNA, which delivers the amino acid to the site of protein synthesis. We know that amino acid is the building blocks for proteins. So we have 5 types of RNA and they are based on their functions. Okay, so let's have the first part, the transcription. Transcription is known as the RNA synthesis. So dito maproproduce yung RNA. So with the direction of the DNA, we are going to produce the hnRNA and mRNA molecules during transcription. And we know that mRNA carries the coded information for protein synthesis. Okay, so transcription. We're going to summarize the process by the following steps for transcription. Okay, so a portion of the DNA double helix will unwind. So we know that unwinding will expose some bases. And this unwinding process is governed by RNA polymerase. So that unwinding, unlike for the DNA replication, ang nagkakos ng unwinding sa DNA replication ay DNA helicase. Dito sa transcription, wala ng DNA helicase, rather, ang magkakos ng unwinding or magdadirect ng unwinding is the enzyme RNA polymerase. And because of this RNA polymerase, magkaiba yung unwinding na nangyayari during transcription at of course, iba rin yung effect ni DNA helicase during the replication. Ang effect ng RNA polymerase unwinding is just partial unwinding. Sinabing partial unwinding, babalik din siya, magre-rewind din siya. Unlike kay DNA helicase that unwinding is permanent kasi nga maggagawa sila ng identical DNA. But for this transcription, the power of RNA polymerase is just partial unwinding. Babalik din sa original structure yung DNA once natapos yung transcription. Okay. And still, there are free nucleotides inside the nucleus, and they are going to form base pairing with this exposed strand kasi nga nag-unwind na siya, so meron ng mga exposed DNA bases wherein a free ribonucleotides. Okay, so hindi na DNA nucleotides ang makiki-base pair dito sa exposed DNA bases, rather it would be ribonucleotides this time. Pag sinabing ribonucleotides, ang kanilang sugar ay ribose. Instead of yung dun sa replication, ang makikibase pair ay free nucleotides with deoxyribose during replication. But during transcription, a ribonucleotides will base pair with the exposed DNA bases that will form new base pairs. Okay, so ito yung sinasabi ko. Uracil rather than thymine will base pair with the adenine. Okay, and only 10 base pairs of DNA template strands are exposed at a time. Okay, kasi nga this unwinding is under the direction of RNA polymerase. Okay? So, ulitin natin, ribonucleotides are involved during base pairing rather than a deoxy ribonucleotides. Okay? And of course, the backbone of the new nucleic acid is having a ribose instead of a deoxyribose. Kasi nga hindi naman DNA ang product during transcription. Ang ita-transcribe natin would be for the synthesis of RNA. That's why we have ribose in the backbone of the new nucleic acid. And we also have RNA polymerase, again, that will involve in the linkages of this new ribonucleotides. Okay, so one at a time in order for a growing hRNA molecule to be produced. So once na nagkaroon ng bagong base pairing, these nucleotides will be bonded together so as to produce the new nucleic acid in the form of hnRNA. Now, transcription ends once RNA polymerase encounters in the bases read a stop signal. So, mag-stop na siya kapag naka-encounter na siya ng stop signal. And the newly formed hnRNA molecules and then the DNA polymerase enzyme are released. Okay, so the DNA then rewinds to reform again in its original double helix structure. So ito yung sinasabi ko kanina, that the unwinding is just temporary because the DNA after transcription will rewind itself to reform into its double helix structure. Okay, so ito yung ating diagram para ma-visualize natin. So, what is needed in the transcription is a DNA molecule. Okay, ito yung DNA molecule. Yes. So, this DNA will have a partial unwinding because of the enzyme RNA polymerase, such that DNA bases will be exposed here. So, may mga adenine dyan, may thymine, guanine, cytosine. So, meron ding mga thymine dyan. Okay. So, may expose na yung mga DNA bases here. And after being exposed, so still inside the nucleus, may mga free ribonucleotides. So, may mga uracil, meron din pa rin mga adenine, and the like. Okay? So, what happens now is, upon exposing this DNA's template strand, the free ribonucleotides will base pair with, will base pair with the exposed DNA strand, DNA bases. So, the adenine, so dito, kunyari, if we have here still the A, T, G, C, and T, okay? So, the free nucleotides, free ribonucleotides will base pair here. So, ito na yung ang makikibase pair with a hydrogen bond. So, A will have U, and then if this is T, this will base pair with A, and then G will base pair with C, C will base pair with, G will base paired with C, and C will base pair with G, T will base pair with A. Okay? So, laging tatandaan, wala nang T, wala nang thymine dito sa transcription because we are producing RNA. Okay, so after that, after this base pairing, these new nucleotides will be bonded together so that an RNA will be produced. So, ito, magko-connecta na sila. Okay? So, this will connect with each other so as to produce the hnRNA. Okay? So, once hnRNA are produced, there would come a time na ma-detect nila yung stop signal. So, mag-e-end na dyan. Okay? And once stop signal has been detected, this hnRNA will be released as well as the DNA strand will reform itself to its original structure. So, ang product natin ay after transcription ay hnRNA and still the DNA on its original structure. Okay, so we have the gene to be the segment of the DNA strand that contains the base sequence for the production of hnRNA or mRNA molecule. And the genome refers to all of the genetic material or the total DNA that contained in a chromosome of an organism. Okay, so we have here, dito sa part na to, yung mga genes. Okay? Now, let's have this base pairing associated with transcription process. So, if we are given with 5' ATGC CA 3', okay, ano ang maproproduce nating hnRNA strand? So, first we get the DNA strand being exposed. So, we have 5' AT GCCA 3'. Sa base pairing will be, so 5', ang hnRNA na maproproduce will start with 3', and A will base pair with U, wala nang T dito, but T will base pair with A, pwede ang A kasi nga meron din naman yan sa RNA. G will be paired with C, C will with G, and A with U. Walang T kasi ang produce natin ay RNA. And of course, this will end with 5'. Okay? This structure could also be rewritten. So the mRNA, the hnRNA could also be written as, so we start here, 5', ito yun, U, G, G, C, A, U 3'. So parehas lang ito, parehas lang. Binaligtad ko lang para mag-start siya sa 5'. Nevertheless, these two are just the same. Pwede itong dalawang to ang pwedeng ma-produce during the transcription. Okay? Another example, so this is 3' TAA CCT 5'. If this is the DNA, the hRNA that will be produced would be 5'. T will be still base pair with A will be with U, G, and A, 3'. Okay, so that would be the HNRA strand that could be produced during transcription. Okay, but the process will not stop there na HNRNA lang ang maproduce natin. We have this post-transcription process wherein the HNRNA, being as a heterogeneous nuclear RNA, will be converted to mRNA. Okay. So this is the conversion of HNRNA to mRNA. Bakit kailangan i-convert? Kasi the portion of RNA has parts na they don't convey genetic information. Okay, so some parts of this Asian RNA are deleted and those that retained are joined together. Kasi ito nga yun, ang Asian RNA being as a heterogeneous, meron siyang exons and introns. Ano ba itong mga ito? Exons, okay? Exons are segments or portion of the gene, or the gene is segmented and it has a portions of exons that contains the genetic information, while other segments known as introns does not convey any genetic information. Kasi nga, HNRNA is a heterogeneous molecule, so hindi siya uniform all throughout. May mga exons na nagko-contain ng genetic informations, pero meron ding mga introns na walang genetic information. So, to produce the mRNA which only bears all the genetic information, kailangan tanggalin itong introns. And that is done during the post-transcription processes wherein HNRNA are being edited by deleting these introns, and the exons that retained are joined together to form one strand. Okay? So, but kailangan tanggalin ang mga introns? Because they may interrupt in the genetic message. Kasi nga kung a portion of the gene may message dito tapos wala, tas meron, tas wala, of course, this information would be interrupted kasi nga wala ngang laman yung introns. Okay? So, an example of this, a gene consisting of exons and introns. So, this is, say for example, the Asian RNA. Very heterogeneous siya. May exon, introns. Okay? Laging tatandaan, exon, merong genetic information. Introns, walang genetic information. So, with the action of snRNA, these introns are being cut and removed. So, tatanggalin daw yan. Tatanggalin. Kasi nga wala naman siyang laman na message. Okay? And still with the action of snRNA, these exons are now being joined together to form the mRNA. Okay, ulitin natin. Exons nare-retain, introns tinatanggal during this post-transcription process. And this post-transcription process is known as splicing. Okay? Okay, so ulitin natin. Ang introns and exons are part ng gene that has been transcribed during the production of hnRNA. Kanya lang, the hnRNA is being deleted under the direction of some enzymes, and the removal of introns and the remaining exons are being joined together to form the shortened RNA. So, syempre, mas lumiit na yung mRNA kasi nga natanggal na yung mga introns. And only the genetic information are being retained on this type of RNA, such that this mRNA or the edited RNA is now the mRNA, which serves as the blueprint for protein. So wala na yung mga walang message. Okay, natanggal na. And sabi ko kanina, that process, that post-transcription process is known as the splicing, or this is the removal of introns from the mRNA and the joining of the remaining exons together to form mRNA. And spliceosome is the large assembly of snRNA. Spliceosome is composed of snRNA that has been attached to some proteins. Okay, so ito yung nagdadirect ng splicing or the removal of introns and the joining back together of the exons. Meron din tayong tinatawag na alternative splicing. This is a process by which several different proteins that are variations of the basic structure motif can be produced from a single gene. So if this is Asian RNA and segment A is an exon, and this blue segment is introns, B is an exon, this is intron, this is an exon, another intron, and an exon. Okay. Alternative splicing is the process wherein mabomodify yung pwedeng ma-produce na mRNA. And because of this modification or variation, ma-produce ang varied or very diversified na mRNA. Okay, so pwede daw na ADB lang, pwede rin na ACD. Okay, the introns will be removed. Pwede rin din na mRNA that will direct the third protein will be A, B, C, D, and pwede rin din na AD. Okay, so as a recap, during transcription, the DNA, under the direction of DNA, hnRNA will be produced. However, this hnRNA has introns or segments that does not convey any message, so they would be removed through splicing, or that is the post-transcription process, to produce the mRNA, which serves as a blueprint for protein synthesis. Okay?